Welcome back, everyone. We have an update from Atara Biotherapeutics, trades on the NASDAQ under the symbol ATRA. They're a leader in the field of allogeneic T-cell therapies and are applying their unique Epstein-Barr virus T-cell platform to develop CAR-T therapies for oncology and autoimmune diseases. Please welcome Head of Corporate Communications and IR, Jason Ah. Welcome back, Jason.
Great. Thanks so much for having me. Hello, everyone. Really happy to be here. And so just as a quick refresher for those that are not too familiar with the story, so Atara is a allogeneic T-cell therapy company. So we're focused on really looking at our unique Epstein-Barr virus T-cell platform to develop the next generation of CAR-T therapeutics. So I'll go through just a couple slides here today. So we really benefit from having a platform that's extremely differentiated in the sense that we use and rely on healthy donor-derived T-cells. And so really what that looks like is for all allogeneic companies, they have to solve for one major challenge is how do you get cells that are different from a patient or host into that host without them being rejected or causing some kind of immune reaction. And so we have a very elegant solution. So we use a healthy immune response to a very common virus called Epstein-Barr virus. And that's probably more commonly known for causing mononucleosis or the kissing disease. And what that does for us is it produces a large amount of these T cells that we're able to extract, manufacture, and then basically store as an inventory. And so what that looks like is if a patient needs it, we're able to get that to that patient within about a 3-day time period in North America. Now, these cells that we use basically solve, like I said, for the main issue or challenge that allogeneic companies face is how you get these cells into the body without it causing some kind of immune reaction. And what that does is that we use Epstein-Barr virus T-cell receptors. And so what that means is it only looks for Epstein-Barr virus and nothing else. And so what that looks like going into a body is instead of having a bunch of different T-cell receptors binding different things throughout the body that can cause graft-versus-host disease, that these Epstein-Barr virus have a very narrow TCR set, and that essentially prevents or mitigates graft-versus-host disease. In the 600 patients that we've treated so far with our platform, we have not seen graft-versus-host disease as it pertains or relates to the drug. Now, the other part of this is how do you keep these cells in the body without them being rejected? And so what we do here is we just do basic transplant biology principles, and we do partial HLA matching. And again, what we've seen is our cells are able to persist and expand in the body. So it's very exciting that we're using this one platform for our portfolio. And actually, our lead asset, tabelecleucel, was approved in late 2022. It's approved in Europe. It's actually the first and only allogeneic T-cell therapy approved so far, which is very exciting. And maybe some of you have seen that we actually just announced that our BLA just got accepted by the FDA yesterday. With priority review. So we have a PDUFA date of January 15th, 2025. So we're very excited about that. And the same platform we're using, that same Epstein-Barr virus T-cell platform underpins as kind of the backbone or chassis of our CAR-T portfolio. And what you can see on the right-hand side here is that we have two assets in that CAR-T side of the house, ATA3219 and ATA3431. So for 3219, it's a anti-CD19 CAR, and it's actually currently in a phase 1 trial for non-Hodgkin's lymphoma. And we're expecting an initial data set later this year in Q4. And we also are pursuing an autoimmune disease. And so we have a phase 1 study that we're expecting initial data in lupus nephritis in the first half of 2025, and that will include lymphodepletion. And what's really exciting is and very differentiating, if it works, is that we're also looking at severe SLE without lymphodepletion. It's a new cohort that we just announced a couple months ago now, and that data is expected in the second half of 25. 3431 is our other asset a little bit earlier in the pipeline. So this is a dual CAR-T targeting CD19 and CD20. And this has actually shown much greater anti-tumor efficacy against autologous benchmark preclinically by us, by Atara, as well as clinically by some other companies that are pursuing an autologous ATA3431. So again, we're pursuing this from an allogeneic perspective with very high objective response rates and complete responses. So again, this is a very exciting asset. This is progressing towards IND submission in the second half of 25. So all in all, I said we have tabelecleucel. This is approved. It's being commercialized through our partners in Europe, a company called Pierre Fabre. They also have global commercialization rights in the US as well. So come early next year, if everything goes correctly, we'll offload this asset. And we'll be left with a double-digit tiered royalty stream moving forward, as well as sales and commercial milestones. And then we're going to be a very focused, company on our CAR-T portfolio of both 3219 and 3431, which again we're expecting data for 3219 for NHL end of this year. And then the autoimmune is a little bit behind in the first half of 25 and the second half of 25 for lupus nephritis and SLE respectively. So we have a very exciting next 6 to 12 months with multiple key catalysts for the company. So with that, that's pretty much the update, and I'd be happy to take any questions.
Great, Jason. All right, so ATA 3219 is unique from the other allogeneic CAR Ts in that you don't gene edit the T cell receptor or MHC. So explain what that is and why is this better?
Yeah, no, good question. So basically, I was kind of alluding to, so all allogeneic companies have to really overcome this allogeneic conundrum, again, of how do you introduce foreign cells into a patient without eliciting alloreactivity or having CAR T rejection. So again, at Atara, we believe that a non-gene edited approach is the best approach. Most other approaches out there really do extensive gene editing to take out the T cell receptor, take out the MHC or HLA. And so what we do is we keep those. And we keep those because we use these Epstein-Barr virus T cells. And ultimately, we believe that this leads to a much healthier, fitter cell than what gene-edited approaches offer, which ultimately has significant cell stress, double-stranded breaks, CMC issues, regulatory challenges, and just really practical manufacturing efficiencies with looking at 3, 4, 5 edits that are 50, 60% success rate. Again, they're kind of multiplicative, and you're left with a very low efficiency from a manufacturing perspective. So again, really believe that no gene editing is better.
And what type of data are you expecting from your phase 1 NHL study later this year?
Yeah, so really this is going to be initial data, on a limited number of patients here that we hope will provide pretty meaningful insights on, initial safety and tolerability. Probably most importantly would be PK/PD, including B cell depletion. So we'll really get a good feel for hopefully figure out how and look at how these cells are behaving and hopefully behaving as we expect them to, and then possibly some preliminary efficacy data. And this NHL data obviously has important implications and read through to our autoimmune programs as well.
And what are the differences between the two cohorts for your lupus phase 1 study?
Yeah, so kind of as I alluded to in the start here, so 3219 is being investigated with in patients with lupus nephritis in combination with lymphodepletion chemotherapy. So that's the first cohort, and really that's just comparing like with Dr. Schett. That's that academic study that came out of Germany last year that really kind of set the field ablaze and kind of moving forward with CAR T. So we wanted to have a like-for-like comparison. But this SLE cohort is going to be without lymphodepletion. So it's really designed to simplify the treatment regimen and hopefully provide a differentiated safety profile for patients that obviously don't want to be on this toxic chemotherapy or lymphodepletion. And importantly, this new cohort does not overlap with the lupus nephritis population, so it allows us to start both cohorts at once, which is really nice.
And talk about your dual targeting CD19, CD20 CAR T. Why pair CD20 with CD19?
Yeah, so it really comes down to, in a lot of kind of tumor environments, as well as hematological malignancies, that CD19 antigen downregulation is really a key contributor to relapse. And it's ultimately just, tumors kind of adapt, right? They evolve and they downregulate CD19. So the hypothesis, the idea behind this is if you can really target two antigens, so CD19 and CD20, that it kind of prevents that even if CD19 gets downregulated, you still have something that's targeting a protein that's expressed on those tumors, which is beneficial. And we've seen that already from a variety of autologous CAR Ts out there that are targeting CD19 and CD20. CBMG's a Chinese-based company, they came out ASH last year, 91% ORR, very good CR rate. So we strongly believe that the targeting both will lead to incrementally better efficacy than just targeting a single antigen. Of course, we have to show that in the clinic, but preclinical data look very differentiated compared to autologous benchmark with much longer persistence and survival. So we're very excited about that asset.
Great, Jason. Do you have any closing remarks for your update today?
Yeah, just appreciate everyone's time. And just as a reminder, we have a very exciting next 6 to 12 months with tabelecleucel and the BLA acceptance that we just announced yesterday and the PDUFA date in January 15th. And then we have a very robust CAR T portfolio of CD19 targeted CAR T for both oncology and autoimmune, and 3431 not too far behind, where we're targeting that IND for the second half of 25. So really appreciate everyone's time, and please reach out if you want to discuss this any further. Thank you.
Perfect. Thank you, Jason. All right, everyone, stay with us. We'll be right back.